Hi, I'm Eric Brooks. I'm a new assistant professor in the Molecular Biomedical Sciences department here at the College of Veterinary Medicine, and I'm an assistant professor of quantitative developmental biology. So I'm really fascinated by how individual cells act cooperatively to change the shapes of tissues. Right? So during the process of organismal development, we start out largely as symmetrical, spherical kind of embryos, and then over time we generate shape, right? And have these arms that are clearly emerging. And so, what's really fascinating is that these big structural changes in the embryo are driven by the individual activities of cells that are coordinately regulated to have this occur. So, the sort of thing that I'm really interested in is so then all these cells have to act together, so they have to be under the control of some kind of spatial signals, right? And so we kind of have three questions that we're interested in. What are these spatial signals and how are they sort of interacting with cells? And then the second question is, what are the cells doing in response to these signals? How is it that they're actually changing themselves, either in their shape or their organization with the tissue to sort of create these larger scale outcomes? And then the final question that I'm really interested in is how do things in the environment, environmental factors, actually alter either the spatial signals themselves or the cellular outcomes resulting from those spatial signals? And we sort of study this in the context of a structural birth defect called neural tube defects. So these are among the most common human developmental disorders. They occur in about 1 in 2,000 pregnancies. And this whole process is actually the conversion of a flat sheet of cells into a closed hollow tube. That tube then forms the structural basis for the brain and the spinal cord. And so if the sheet fails to convert into the tube, you have what's called the neural tube defect. And that's kind of where I'm interested in studying these kind of cellular questions at the moment.